You're welcome to this YouTube channel once again. For new viewers, please kindly subscribe and don't forget to click the notification bell icon so you get notified each time a video drops on this page. And also don't forget to check out our previous videos on comparative anatomy and also surface anatomy of upper limb. Today we'll be looking at tissue sampling acquisition techniques under microscopy. Let's get right into it. Introduction. The tissue undergoes series of steps before it reaches the examiner's desk to be thoroughly examined microscopically to arrive at a particular diagnosis. Therefore, to achieve this, it is important that the tissue must be prepared in such a manner that it is sufficiently thick or thin to be examined microscopically, and all the structures in the tissue may be differentiated. A tissue, as we all know, it's a group of cells specialized and differentiated to perform a specialized function. What are the differences in tissue sample acquisition techniques? The first one we have here is the fine needle aspiration. The aspiration of tissue sample with a needle size smaller than 21 gauge is considered as a fine needle aspiration. For the purposes of endoscopic ultrasound-guided fine needle aspiration biopsy, for a long time 22 gauge or 25 gauge and now 27 gauge needles have been used to obtain samples from various organ sites. Also, using such thin needles have proven to be less invasive and has the advantage of obtaining adequate samples from target lesions to provide an accurate diagnosis. The other one we have is the core needle biopsy. The core biopsy needles, they allow acquisition of tissue core biopsies. Innovation has allowed acquisition of core biopsy samples with a needle size as small as 25 gauge, 22 gauge and 19 gauge. Also, a sample acquired using this modality offers core biopsies and the advantage of reviewing tissue architecture. Such a sample often helps to reduce the number of passes. Then, it also helps general surgical pathologists who are more familiar with such samples to render their opinions. Types of materials obtained in laboratory. As biopsy, a small piece of lesions or tumor, which is sent for diagnosis before final removal of the lesion or the tumor. If the whole of the tumor or lesion is sent for examination and diagnosis by the pathologist, it is called the excisional biopsy. Then tissues from the autopsy are sent for the study of disease and its cause for the advancement of medicine. Now, there are three types of histological preparation. We have the whole mount, the sections and the smear. Looking at the whole mounts, there are preparation of entire animal, e.g. fungus, parasites. These preparations should be no more than 0.2 to 0.5 millimeter in thickness. Then for sections, the majority of the preparations in histology are sections. The tissue is cut into about 3 to 5 millimeter thick pieces, processed, and 5 micron thick sections are cut on a microtome. These are then stained and permanently mounted. The microtomes, they are special instruments which have automatic mechanism for cutting very thin sections. Then to cut the sections on the microtome, the tissue must be made hard enough to not get crushed. There are two methods of hardening tissue. One is by freezing them and the other is by embedding them in a hard material such as paraffin wax or gelatin. Then for smears, they are made from blood, bone marrow or any fluid such as pleural or acidic fluid. These are immediately fixed in alcohol to preserve the tissue cellular structures, followed by staining. Then smears are also made by crushing soft tissue between two slides, or an impression smear is made by pressing a clean slide in contact with the moist surface of the tissue. Now, what are the responsibilities of a technician? The technician is responsible for specimen preservation, specimen labeling, logging, and identification, and also preparation of the specimen to facilitate gross examination and microscopy. And lastly, record keeping. To obtain these aims, the following points need to be considered. As soon as the specimen is received in the laboratory, you check if the specimen is properly labeled with the name, age, hospital registration number, and the nature of tissue to be examined. Also, you ensure the requisition form is also duly filled. Also, you check if the specimen is in proper fixative. Fixative should be 15 to 20 times the volume of the specimen, and fixative if not present insufficient. Add fixative if not present in sufficient amount. Also, you check if the financial matters have been taken care of. You also make the entries in biopsy register and give the specimen a pathology number called the accession number. You note this number carefully on the acquisition form as well as the container. This number will accompany the specimen everywhere. If the specimen is large, inform the pathologist who will make cuts in the specimen so that proper fixation is done. And also, the container should be appropriate to hold the specimen without distorting it. You have to also blocks of tissues taken for processing should be left in 10% formalin at 60 degrees Celsius till processing. These would then be fixed in 2 hours. The slides should be released for recording after consultation with the pathologist. Also, specimens should be kept in their marked container and discarded after checking with pathologists. Then blocks must be stored in their proper number the same way. Note, the blocks have to be kept preserved for lifelong. Then slides should be stored in their proper number after 3 days. It gives time for slides to be properly dried. This is the end of this lecture. Thank you for listening. Hope to see you in our next lecture. Goodbye for now.